Hi guys, now let's learn about the M phase. The M phase consists of two stages. The first one is the division of the nucleus, or known as karyokinesis, followed by the division of cytoplasm, also known as cytokinesis. Now let's learn about karyokinesis first. Karyokinesis, which is the division of the nucleus, really defines the type of the cell division. There are two types of cell division, which is mitosis and meiosis. Mitosis is the type of cell division whereby the two daughter cells produced will be genetically identical and have the same chromosome number as the mother cell. Meiosis, however, refers to the type of cell division whereby 4 daughter cells are produced, and these daughter cells are not genetically identical and have only half of the chromosome number of the mother cell. Let's focus on mitosis first. There are 4 stages to mitosis: Prophase, Metaphase, Anaphase, and Telophase. Let's learn about each of the phases. Prophase. Inside the nucleus, the decondensed chromosome in the form of chromatin will begin to condense or coil, as you can see in the diagram. Each of the individual chromosomes can now be seen clearly because they appear thicker and shorter. Each of the individual chromosome has been duplicated during the S phase. This is before replication, and this is after replication and being condensed. In this form, the chromosome is known as sister chromatid. This is because this chromatid is exactly the same copy as this chromatid. The sister chromatids are attached at this region called the centromere. As the chromatin condense into individual chromosome, the nucleolus disappear, as you can see here. Besides that, the nuclear membrane, labeled purple, will begin to disintegrate, and the centrosome containing centrioles will begin to migrate to opposite poles, radiating spindle fiber. Now let's take a look at a video showing how the DNA is packed, forming sister chromatids during prophase.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Next is the metaphase stage. During metaphase, the centrosome has arrived at the opposite poles. The sister chromatids align at the metaphase plate. Each sister chromatid is attached to spindle fiber from both poles at the centromere, specifically at a region called the kinetochore. It is important for the students to note that since centrioles can only be found in animal cells, so therefore the structure here, called the ester, can also only be found in the animal cells. The next stage is the anaphase. During anaphase, the sister chromatids split at the centromere, as you can see here. Then the spindle fiber will shorten or contract, so therefore each chromatid produced from the splitting of a sister chromatid will be pulled to the opposite poles, just like this. The last stage of mitosis is the telophase. In telophase, each chromatid has now arrived at the opposite poles, as you can see in the diagram. As the chromatid arrive, they begin to decondense, so therefore making you unable to see individual chromosome anymore. In addition to that, as the chromatid decondense into chromatin, nucleolus will reappear, and nuclear membrane will also reappear. As you can see in the diagram, telophase is quickly followed by the division of the cytoplasm, or also known as the cytokinesis. We will learn about cytokinesis next, but before that, let's watch a video summarizing mitosis.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Cytokinesis refers to the division of the cytoplasm. In animal cells, it happens when the actin microfilament that can be found on the surface of the plasma membrane facing the cytoplasm begin to contract inward. This will cause a ring of the plasma membrane to be pulled inward, as you can see here. This is the actin microfilament that can be found on the surface of the plasma membrane facing the cytoplasm. As the actin microfilament begins to contract, it will pull the plasma membrane inward, forming a cleavage furrow. As the cleavage furrow deepens, the cells will separate into two independent daughter cells. In plant cells, however, the formation of the cleavage furrow is prevented by the rigid cell wall. To perform cytokinesis, plants need to deposit new cell wall in between the two daughter cells. Vesicles containing the new cell wall materials comes from the Golgi apparatus, as you can see here. These vesicles will be deposited in the region between the two daughter nuclei, forming a cell plate. As more and more cell wall material being delivered, the cell plate grows outward until the membrane of the cell plate fuses with the membrane of the mother cell, separating the two cells.